So I'm a researcher at the South Australian Museum. We do a lot of research at the museum, which is my primary task. Work on Collembola and Hymenoptera mainly, but also on Lepidoptera. Everybody has to work on it to make Paul happy. And we've got a few projects along that in mostly Australasia, the Pacific, Antarctic and subantarctic. Ian Hogg and I were on a big cruise around Antarctica. We did the Hobart to South America leg. So we've got a whole lot of material there waiting to be barcoded. So we have to get on top of that. Now, because I've sort of recently come into being the rep for Australia, I'm not really on top of who's barcoding in Australia, but there are a lot of people doing work, just not in any sort of coordinated way. But as you can see, we've got a large number of sequences. The flashy map with all the colours on it shows Australia is participating, but obviously that can be done in a much more coordinated way. And of course, like usual, arthropods make up the largest contribution to the BINS species. We have had efforts in Australia to have our iBOL, or contribution, coordinated efforts, but this went on for a few years and we had meetings, but essentially sort of has gone nowhere. Basically, because they've been trying to find funding, big funding, people would promise, it looks promising, and then falls in a hole because they want to spend money elsewhere. But I think funding in Australia the big funding is very hard to get, but there are lots of small funding. We've run, well, since I've been back in Australia for the last 10 years, we've had enough money to do research projects every year, just from small funding opportunities. They fund students, they fund research to do pretty much anything we've been wanting to do. Obviously, getting salaries for people is a big shortfall. So, some of the projects, because, as I said, new to this, but so I've just targeted some of the projects that I'm involved in primarily over the last few years. One of those that we're actually doing at the moment is the idea of barcoding all of the Hymenoptera and particularly the native bees in Australia. So this is underway, have funding for this, so that part of it's not an issue. The issue is getting the specimen, putting it in a leg in the well and sending it. But we're coordinating with Curtin University recently, so I've got a student there that's going to plate up, I think, 3 or 4 plates. So we've got at least 200 species. Just from one institution. We're doing the same. We've got another 200 or 300 species. So this is well underway. And with iBOL 2, it was the perfect time to ramp that up and actually make it happen, rather than just thinking about it and doing it in little steps. Another story from, again, native bees, but this time in the Pacific, Fiji, where we have had students working on, so we've been working in Fiji since about 2010, and Fiji has 4 endemic bee species. We're now up to 26, so it's not a lot, but it's a lot more than was known, and they're all morphologically identified. They're actual species, whichever way you look at it. But the interesting thing, and this comes back to what Paul was talking about, the biosurveillance, because 22 of the 26 species are only found above 800 metres in elevation. And so they've only got, the highest mountain I think is 1,300 and something metres. They've only got 500 metres to play with before they disappear off the top. And as you can imagine, the climate above 800 metres in Fiji is very different to the lower part. You think of the tropics, the top above 800 metres, you wouldn't know you're in the tropics. So it's very much drier and it's much cooler. And nighttime temperatures can get down to 4 degrees. Yet when you're in the lowlands, the nighttime is basically the same as the daytime. So it's a distinctly different climate, these species are in danger of going extinct. We've recorded one going extinct already, collected by Michener back in 1960s, described in the 70s. From that record, we went back to exactly the same site. We collected a male and a female. That was back in 2012. We haven't recorded it anywhere since. So this was at 1,100 metres. So it's basically hit its thermal tolerance, it's disappeared. And this is after a lot of sampling in the highlands. Another project that we're doing, what have I written there? Lepidoptera. So again, keep Paul happy. We've been working with a few people overseas, and Thomas Simonson. And this is underway. Again, we've got some funding, small funding efforts in South Australia. So these are just student projects, or anybody can apply for them, but distinctly almost easy to get. They give money away. It's really weird. They actually email you and say, please put application in this year. We would like to give you money. But it's enabled us to do a number of projects, mainly on the Hepialidae, Castaneae, and some of these published, but just simple, these are students or honorary researchers at the museum who have an idea. I say, yeah, sure, let's do it. But it adds to the whole plan, sequences, etc. Another project, salt lake fauna, which is relatively understudied in Australia. So these are animals, unlike some that can't live on the salt lakes, there are others that can. And scorpions, beetles, crickets, they're all endemic to each lake, basically. And so we've sampled around, and again, people that wanted to pay for it give us funding, so we've been able to do a fair bit there. And we find, actually, let's go back. So we targeted 4 main groups of the main animals that you'll find on the lakes. And so this is just an example of one of those. It's a scorpion, only one species in the genus. And we've got about 6 species across, just in South Australia, from that work. And that's corroborated by nuclear data as well. Haven't looked at morphology. So this SWOT analysis, I didn't really know what to do here. Strengths, I think, because Australia is like we do a lot of research, like most countries, and people are doing a lot of barcoding, whether they know it or not. So it's just a matter of just being coordinated, and there is funding out there. So obviously, I think that's the main strength and weakness of Australia is, it's there, it's just a matter of making it coordinated. The threats, obviously no money, but I don't see that as for our being able to contribute to iBOL 2, I don't think that's an issue. I think we can do it. Opportunities, I think, and Paul mentioned this, like PNG with being a member or a associate member, and I think that's one thing that Australia could do more of, is more work in other countries. Like we've been in Fiji since 2010. We've also been Samoa, Solomon Islands, and other places. So we have connections there, and I think fostering that as a member of iBOL 2, I think that would be a role as well in the neighbouring countries, helping in whatever way we can, and then the Pacific neighbours. I think that's all I want to say. Thanks.